is Krebs? Krebs is the son of a Jewish surgeon. He was born in Germany in 1900. He researched at a variety of places. He actually fled Nazi Germany in 1933 and went from the University of Freiburg to the University of Cambridge. In 1953, he won the Nobel Prize for his discovery of the citric acid cycle, which is the Krebs cycle. And he wrote Energy Transformations in Living Matter and helped co-author Reminiscence and Reflections.
So the Krebs cycle is highly important, and it serves many important purposes. Firstly, it breaks down all metabolites like sugars, fatty acids, and amino acids. It uses 8 different enzymes to transform acetyl coenzyme A into molecules of carbon dioxide and 1 molecule of ATP. It also prevents the waste of metabolic energy that would occur if the cycle were to run continuously. So it increases the cell's ATP producing potential, and it's the driving force of cellular respiration.
So now the reactants and products. So what do you need for the Krebs cycle? So where does this all happen? So the cells that have the Krebs cycle or cellular respiration are aerobic bacteria, free-living protozoa, most algae and fungi, and most importantly, animal cells. That's the one we're most familiar with. More specifically, in eukaryotic cells, it's happening in the mitochondrial matrix, which is basically the cytoplasm of the mitochondria. It's actually a lot less liquid than the cytoplasm of the cell, and it has the DNA and ribosomes of the mitochondria, but most importantly, it has the enzymes that are going to catalyze the oxidation of pyruvate. And then in prokaryotic cells, it's all happening in the cytosol, because prokaryotic cells don't have mitochondria, so it's going to happen there. And the cytosol is basically the cytoplasm that's not bound by organelles.
We're actually going to run through the steps going down from glycolysis through the prep step and into the actual Krebs cycle. I'm going to run through glycolysis really quick just because it leads you into the prep steps. This diagram right here going down is your progression of events, and from left to right is getting put in, like the ATP you can hear, 2 ATP comes in and 4 ATP comes out, so anything on the right side means that it actually is getting produced. A column showing things that are produced so we can keep track of that, see what our net production is in the end, and on the left over here I show where each step begins and ends and where it actually occurs. Running through glycolysis quickly, pretty much what happens is you have a glucose with 6 carbons right here. It gets split into 2 different pyruvates, each which have 3 carbons. We have a net of 2 ATP production and 2 NADHs. Pyruvate that was created in glycolysis is going to enter into the prep steps and into the Krebs cycle. Because 2 pyruvates are created here and here, this whole process is actually going to happen twice. So for the products, I have for one and then for both of them, so you can keep track, try to have it be a little bit more clear. So our 3-carbon pyruvate, 1 carbon gets busted off in the form of CO2, so 1 CO2, 2 over here. NADH, or NAD I should say, gets reduced into NADH, and this forms acetyl-CoA, which is a 2-carbon, because you got that 3 minus 1, 2, and from there it's actually going to enter into the mitochondria. Mitochondria, acetyl-CoA is going to merge with oxaloacetic acid, which is a 4-carbon, so you have the 2 and the 4, and you're going to get citric acid, a 6-carbon molecule. And the rest of these steps are actually just going to be a series of oxidations which turn citric acid back into oxaloacetic acid that can then merge again with acetyl-CoA. We start out with NAD that gets reduced into NADH, happens again right here, and then we have 2 carbons that are broken off and that forms 2 CO2s, you can see right over here. in between all of these different steps, it does form different carbon molecules, but those carbon molecules exist so that we can reduce these molecules and use them later on in your electron transport chain. Following the carbons being broken off, we have an ADP, which becomes ATP, gaining another phosphate group, and FAD also gets reduced into FADH over here. This happens once, and again we have an NAD into NADH. And then after that, you lost those 2 carbons, so 6 minus 2, you get right back into your 4 over. Looking at our net production for each of these steps, for glycolysis, which happens in the cytoplasm, we get 2 ATP and 2 NADH. Once we move down into the prep step, because it occurs twice, we're doubling the amount that we get from one single prep step and Krebs cycle. So we're going to get a total of 2 carbon dioxides and 2 NADH from the prep step. Moving down into the matrix of the mitochondria, we're going to get 4 NADH, 4 carbon dioxide, 2 ATP, 2 FADH2, and an additional 2 NADH. We're going to get a total of 10 NADH, 2 FADH2, and 4 ATP. So it's important to note here that the Krebs cycle, its purpose directly is not actually to create ATP, because as you can see, you're only getting 4, 2 of which are directly from the Krebs cycle. The goal is to create all of these other byproducts, NADH and FADH2, which can be used in the electron transport chain to produce ATP. So those 10 NADH become 30 ATP later, and the 2 FADH2 become 4 ATP later. So as you can see, we have a net of 38 ATP, which is going to be your ideal amount. It's realistically not going to happen most of the time, but that's what would happen if everything theoretically went perfectly.
Alright, some fun facts about the Krebs cycle is it happens in the matrix. No, not the movie The Matrix, unfortunately, but in the matrix of the mitochondria. This cycle does not consume any ATP molecules, and vitamin B is essential in the building blocks for what is needed in the Krebs cycle, specifically vitamin B2 and B3. Here we go. And here are our sources.